All right, let's talk about a technique that totally shook up the world of genetics back in the 90s. It's called Random Amplified Polymorphic DNA, which is a mouthful, I know, so we just call it RAPID, or RAPID. And honestly, RAPID is the perfect word for it. It was this incredibly fast, simple, and super cheap way to get a sneak peek into an organism's DNA. It just blew the doors wide open for scientists everywhere. So let's get into how this really clever technique actually worked.
Okay, to really get this, I want you to imagine something. Let's say you're a map maker, a cartographer, right? But you've got no satellites, no aerial photos, you don't even have a good compass. All you've got to work with are a few random, scattered landmarks. So how on earth do you build a map from just that? Well, believe it or not, that's pretty much the exact challenge geneticists were up against before the 1990s. They were trying to navigate these huge, unsequenced genomes, and it was like trying to map a country in the dark. And that right there, that's the heart of the problem. We can call it the geneticist's dilemma. See, they knew there were incredible secrets, real treasures hidden away in this vast landscape of DNA. The problem was they just didn't have the tools to draw the map to find them.
So back then, the big, fancy technique everyone used was called RFLP. It was considered the gold standard. But here's the catch, and it was a big one. It was painfully slow, super expensive, and it had this major roadblock. To even get started, you had to already know part of the DNA sequence you were studying. Think about that for a second. For countless species out there, especially in plant and animal science, that information just wasn't available. It was a classic catch-22. But then, in 1990, something new came along. A solution popped up that was about to change everything. A technique so accessible, so fast, that its name, RAPID, was often just pronounced rapid.
So, what exactly is RAPD? Well, at its core, it's a really clever spin on a process you might have heard of called PCR, the polymerase chain reaction. Now, with normal PCR, you design these specific little starters called primers to go find a gene you already know about. But with RAPD, you just throw that whole idea out the window. Instead, you use a single, really short primer, we're talking maybe 10 DNA letters long, with a completely random sequence. This little primer is kind of like our lost map maker, just wandering through the genome and landing on any spot that happens to be a match. And here's the absolute game changer. You don't need to know a single thing about the DNA sequence beforehand. It just works.
So this thing just explodes onto the scene in 1990. And get this, it was developed by two different labs totally independently at the exact same time. It was an instant hit, for real. Labs all over the globe jumped on it because it was so fast and so cheap. But, by the mid-90s, a pretty big crack started to show. The results, well, they weren't always consistent. Even with that flaw, though, RAPD's influence was massive. It really paved the way for a lot of the more modern tools we rely on today. Okay, so how does this whole random copying thing actually work? Let's get into the nitty-gritty of the process. The easiest way to think about it is like a three-step dance that's all about temperature, and we just repeat it over and over. First, step 1, we crank up the heat to about 94 degrees Celsius. This is called denaturation, and it basically splits the two strands of the DNA helix apart. Then, step 2, we cool it way down to around 36 degrees. This is the annealing step. It's when our short, random primers can go find a spot on the DNA that they match with and stick to it. And finally, step 3, we bring the heat back up to 72 degrees. That's the sweet spot for an enzyme called Taq polymerase to come in and start copying all the DNA that's sitting between two of those primers.
Okay, now this is where the real magic happens. So the amplification, that massive copying of DNA, it doesn't just happen anywhere. It only kicks off when you get a bit lucky. You need two of these random primers to land on opposite strands of the DNA facing each other, and they have to be close enough for the enzyme to bridge the gap. When that perfect random combination happens, bam, the whole chunk of DNA between them gets copied, and you repeat this cycle, right? So with every single cycle, the number of copies doubles. You go from 2 to 4, 4 to 8, and pretty soon you have millions and millions of identical DNA fragments.
So after you run about 40 of these cycles, you've got this, well, it's basically a soup full of DNA fragments of all different sizes. You take that and run it through an agarose gel. It's kind of like a thick jello. You apply an electric current, and the DNA starts to move. The little tiny fragments can zip through the gel really fast, so they travel farther. The big, bulky ones get tangled up and don't move as far. Then you stain the gel, and what you see is this unique pattern of bands. It looks just like a barcode, and that barcode is a genetic fingerprint.
If you compare two different individuals, their DNA sequence will be slightly different, right? So the primers will bind in different spots, creating a totally different pattern of bands. It's a visual way to see genetic differences.
So you can see this technique was seriously powerful. But like we mentioned earlier, it wasn't perfect. Not by a long shot. Let's dig into both sides of the coin here, the power and the pitfalls.
And I mean, the applications were just huge. Because it was so easy and cheap, scientists started using RAPD for everything. Seriously, they were making the very first genetic maps for species nobody had ever studied before. They were looking at genetic diversity in wild populations, finding genes for disease resistance in crops, even using it for DNA fingerprinting in breeding programs. Basically, if you had any question at all about genetic differences, RAPD suddenly gave you a fast and easy way to at least start looking for the answer.
This is where you really see the trade-offs, right? Let's put it all on the table. In the advantages column, you've got stuff that was absolutely revolutionary. It was fast, it was cheap, and you could use it on pretty much any organism you could get DNA from, all without knowing a single thing about its genome beforehand. Amazing. But the disadvantages column was pretty heavy too. The number one problem was reproducibility. That low temperature we talked about, the one that let the primer stick randomly, it also meant that the tiniest change in your lab setup, the temperature, the chemicals, could give you a completely different result. Another huge issue is that these markers are what we call dominant. What that means is when you see a band on the gel, you know that piece of DNA is there. What you don't know is if the organism got that gene from one parent or from both. You can't tell the difference between a homozygote and a heterozygote. And for some types of genetic studies, that ambiguity is a really big problem.
And this quote just hits the nail on the head. Notoriously laboratory dependent. That says it all. You could do an experiment in your lab in California and get one set of bands, and a researcher in Japan doing the exact same experiment could get a completely different result. Heck, you could get a different result in your own lab just by doing it on a Tuesday instead of a Monday. And you can imagine this made it incredibly difficult for scientists to compare their data or build on each other's work.
So with all these pretty serious problems, you might be wondering, what is RAPD's legacy? Why do we even still talk about it today?
Well, its legacy is all about one word: access. You could say that RAPD democratized genetics. For the very first time, you didn't have to be at a huge, well-funded institution to do this kind of work. Small labs, graduate students, researchers in countries with limited resources, suddenly they could all perform powerful genetic analysis. They didn't need a massive budget, and they didn't need a whole library of existing DNA data. It really truly kicked off this global wave of discovery and empowered a whole new generation of scientists.
And it wasn't just a dead end either. Scientists got clever. They figured out a way to tame RAPD's wild random nature. What they would do is find an interesting-looking band on their RAPD gel, literally cut that little piece of gel out, and then figure out the exact DNA sequence of that fragment. Once they had the sequence, they could design longer, much more specific primers that would only amplify that one single region every single time. This created a brand new type of marker called a scar, and it was basically the best of both worlds. You use the random power of RAPDs to discover it, but then you turn it into a reliable, reproducible tool.
And all of this kind of leaves us with a bigger question, doesn't it? The whole story of RAPD is a perfect example of a trade-off in science. No, it wasn't perfect. It definitely wasn't always reproducible. But you know what it was? It was fast, it was cheap, and it was accessible to everyone. It let thousands of scientists start asking questions they simply couldn't have even tried to answer before. So it makes you think, in this constant race for scientific discovery, when is a tool that's just good enough actually better than one that's perfect?